Okay, good morning, everybody. Good morning. Can you believe we only have this week and next week, and then the class is over? I can't believe it. Yep, time has flown. I'm going to pull up the PowerPoint. We got to take kind of a brisk walk today. We need to finish up what we started last class, and then we also need to probably jump in to the next lecture as well, because we're just running out of time. So let me share this. You guys should have seen that I posted a extra credit assignment on Blackboard, which I never do extra credit for this class ever. But this was just too good of an opportunity to pass up, given the fact that well, first of all, it's we're still dealing with things being very virtual these days, and so if we were not in this situation, normally would have been kind of unrealistic to ask you guys to go all the way to the Bronx to go to that that seminar. But since it's virtual and the content is very similar to stuff that we have been talking about, it's just going to be great. So I'm really excited. It's also given by a PhD student, so it'll be nice to give that student some audience, because I've presented at the Torrey Botanical Society over the years, and even in the before times, it was very hard to get people to go to the Bronx. And I remember, I think I had an audience of like 20 in this giant auditorium. It's very overwhelming. Okay. Hopefully you guys can see the PowerPoint slide. I'm going to pull it up big. Last time we kind of covered cells, right? We covered vessel elements, so xylem tissue, phloem tissue. That's what we covered last time. And just as a reminder, at the end of this lecture, there's a nice summary of all the different cell types and their characteristics at the end of this lecture. But let me pull this up big. All right. So we gotta move. Yeah. So we know we're in the angiosperms, right? We're in the angiosperms, which is the biggest component of the plant kingdom, right? Most of the plants in the plant kingdom are in the flowering plants. Most of them are, right? There's almost 370,000 species of flowering plants, right? So what that means for us, when you're dealing with such a great number of species, is that you end up with a ton of diversity in flowering shape and form, but also in plant shape and form. This slide, I think we've maybe seen before, just gives you some idea of the diversity that we see in flowers in the flowering plants. But obviously there's not 366,000 flowers on this slide, right? So this is just the tip of the iceberg here in terms of the diversity. So let's talk about some of that diversity. Well, so we learned in the conifer lab that the tallest trees that are live on the planet are in the conifers, right? But there are still some pretty tall angiosperms as well. The tallest angiosperms that exist are these eucalyptus trees that are located in southern Australia. Their common name is mountain ash, not to be confused with the ash trees that are being subjected to the ash borer. Totally different thing, but still. And these trees can get really, really tall, about 330 feet tall and about 70 feet wide. So that's like a massive, massive tree. So these are the largest angiosperms. Angiosperms can also be really, really tiny, right? So these are plants in the family Lemnaceae. That's the name of the family. Collectively called the duckweeds, not to be confused with the duckweeds that we learned about in the fern lectures. This is, again, I think I even mentioned it then, why common names are sometimes not super useful, because people use common names in different ways and sometimes describing totally different organisms, right? This is a good example. We had a duckweed that was a fern, and now we're talking about duckweeds that are in the angiosperms. So the duckweed that was a fern, right? Those were like your azolas, right? See, it says it, not to be confused with azolla, a fern, right? Ferns don't make flowers, totally different thing. But these Lemnaceae are also super, super tiny. You can see on the top left picture is a picture of a bunch of them. On the bottom, you see a bee, a bee actually like walking on a mat of these tiny plants. So that's how small they are. They also, like azolla, they also grow in aquatic environments and can form these like dense mats. And if you look at the bottom right, this shows you each of the dots on the map on the bottom right is actually a recorded Lemnaceae occurrence. So this species or this family of plants has kind of taken over worldwide distribution in freshwater bodies. In part, it's thought in part because of the aquarium industry, but also because these plants are so super, super small. When plants, sorry, got plants on the brain. When ships travel around the world, it's very easy to like, you know, accidentally pick up a little bit of this and vector it around in their bilge tanks. So super, super small. Look, only 1 millimeter wide. That's small. Look, I mean, a bee, look at the bee standing on all of that. Okay. Now, oh yeah, you might look at this. You might look at this top left thing and say to yourself, oh, Catherine, what's the topic? The topic is we're finishing up the Angios. So we have 3 total angiosperm lectures. This is technically, oh, sorry, I meant the Tory talk for extra credit. The description is on Blackboard, but the topic of the Tory talk is about the development of the seed coat in certain members of the gymnosperms. Yeah, so we'll be talking and also a little bit about the flowering plants seed coat as well. Yeah, but there's a full description on Blackboard. Sorry, I had just seen the chat box. It didn't pop up before. I don't know why. This is technically still lecture 19. This is the second half of lecture 19, but we will hopefully start lecture 20 today. Either way, just keep in mind for the last exam, it's going to be all the angiosperms. So it doesn't really matter what number they're in because they're all going to be on, anything with the angiosperm is going to be on that last exam. All right. So when I show you this, and I tell you it's a flowering plant, right? Like this is a flowering plant, right? All of these little dots in here are like flowering plants. You might be like, yeah, right. Like how could a flower even fit on one of these little tiny plants, right? Well, it can and it does. Can and it does. This is a close-up of one of those Lemnaceae duckweeds. And you can see each plant produces one incomplete flower. So we learned about complete and incomplete and perfect and imperfect flowers, right? So this flower is incomplete because it has no petals and no sepals. When we say something's incomplete, we say that it's missing what? What is an incomplete flower missing? Yes, Brittany, a whorl, a whorl. If you're an incomplete flower, you're missing a whorl. Remember, there's 4 whorls that flowers have, right? Sepals, petals, stamens, pistil. If one of those is missing, you're an incomplete flower, right? And in this case, this flower has no petals or sepals, so it's missing 2 whorls. However, this flower contains male and female sex organs, and so how would we describe this flower if it contains male and female sex organs? There's like a couple ways you can think about it. So note in the top, it says it on the slide, guys. Each plant produces one incomplete and perfect flower, right? Perfect flower because it has male and female sex organs, right? Okay, you guys haven't woken up yet. I get it. But wake up, let's go. Here's a picture on the left. This is the plant on the left, and you can see 2 stamens and 1 style, 2 stamens and 1 style. So there I have the stamens circled in red, and then that little thing down there is the style. So believe it or not, that entire thing, those 2 stamens and that 1 style is 1 flower. No petals in this case, no sepals in this case, but still a flower. And here's a recall slide, so you can remember these terms, right? Complete versus incomplete, perfect versus imperfect. We've talked about that before. All right. Angiosperms are monophyletic. They are very diverse, right? They have a lot of different habits. They have a lot of different habitats, different sizes, different forms, all right, morphology. But there are some features that they all share and that are unique to the angiosperms. So we say that angiosperms are a monophyletic group in that they're all derived from a single common ancestor. So here you can see this is like a tree, not like a plant tree, right? Like a lineage tree, right? Phylogenetic tree, showing you the relatedness of the members of the angiosperms. This box has inside of it all of the angiosperms. And this blue dot is showing you that there was some common ancestor in evolutionary time that they all are derived from. They're all derived from. So if we're to look at the different types of plants inside of the angiosperm, so everything on this slide, all of these names are types of angiosperms. And you may have remembered that the very beginning of this lecture last time, I mentioned we call the angiosperms Magnoliophyta, right? Magnoliophyta. As versus the older Anthophyta, which we'll talk about in a couple slides. But most of the, so all of these things here are classes. Well, all of the green things here are classes. Don't worry about that right now. The point is that most of the things here only have a few members. So everything that's green on this slide, there are not that many plants within those green things. Each of those green things doesn't contain that many things. But the black things here, the monocots and Dicots, are what contains most of the plants that are in angiosperm. See, so there's 2 classes that comprise the majority of the species, and those are Eudicotyledone and Monocotyledone. Because that's a mouthful. Because that's a mouthful. We just say Eudicots and Monocots, right? Instead of Eudicotyledone and Monocotyledone, even though Monocotyledone kind of sounds like a great stage name, but no, we just say Eudicots and Monocots. And so most of the species are in those, the Eudicots and the Monocots. Here are some examples of what they look like, right? So here we have some Eudicots. Eudicots have about 200,000 species in them. So remember, the angiosperms, there's about 366,000 species, right? And of those 366,000 species, 200,000 of them, 200,000 of them are Eudicots. So that's the very large, right? The largest group of the angiosperms. And of course, if it has 200,000 species, it's going to be very diverse. You have trees, shrubs, herbs, etc. Some examples here, we see a cacti is a type of Eudicot. This little flower called anemone and California poppy. These are just examples. I pulled 3 out of 200,000, right? Here's 3 examples. And then you have the monocots. Monocots have a lot of species as well, right? 90,000, that's a lot. And contain plants you might be familiar with, including grasses, lilies, orchids, cattails, palms, bananas, and rice, right? So those are examples of plants in the monocots. So this is that history I was telling you before, right? So back in the day, we used to call the angiosperms Anthophyta because as botanists, we thought there was only two classes. We thought there was only the dicots and the monocots. That's just what we thought. And so it was really nice and neat. We have dicots, we have monocots. We're going to call them Anthophyta. And basically the way we had to, back then, the way things were decided is we basically had monocots, and then anything that wasn't a monocot, we just said it's a dicot. That's kind of how it worked back then. You have to keep in mind, this is before there were things like, you know, DNA analysis, and people were just going based on traits and physical appearance and like that, those kind of things. So yeah, so it was like we have monocots and then everything else is in a dicot. That's what we used to do. But right, so this is how it used to look. Everything in red used to be within the dicots, and everything within blue was the monocots, right? And you may see in lab, you will very likely see the terms monocot versus dicot, and that's okay. I'm just giving you the context of why that's a oversimplification and it's not quite accurate today, but for the purposes of lab, monocot and dicot are fine. But now we know that dicots are way more complicated. They are just complicated. It's not as simple as monocot and dicot anymore. And so the phylum Anthophyta was redefined and broken up into more bits, such that we had monocots, eudicots, or the true dicots, and then everything else got lumped into what's called basal angiosperms. So it's just like everything else. And those everything else don't really fit neatly into either the monocots or the eudicots. And it's kind of like, I don't know if you ever watched Gilligan's Island, but it was like, you know, and his wife, movie star, and the rest. It's kind of like the basal angiosperms are kind of like, and the rest, it's just the rest. Not monocot and not eudicot. Right. But even plant people still use the term dicot. It's just out of habit. And when a plant person says dicot, they mean eudicot. Just keep that in mind. In lab, we still use dicot. But when you say dicot, it's just assumed you mean eudicot, FYI. And so now angiosperms are Magnoliophyta and not Anthophyta anymore. All right. Let's talk about flowers. So when you're comparing the eudicots or dicots and the monocots, there are ways to tell when you're looking at the plant and the flower on the plant. There are ways to tell if you're looking at a monocot or a eudicot. So eudicot or dicots have flower parts arranged in 4s and 5s. So their whorls contain 4 or 5 things, whereas monocots have their flower parts or whorls in 3s. What do I mean by that? I'll show you. I will show you. Let me use blue. All right, so on the top here is an example of a flower, and look, it says, so this flower has 5 petals, right? 1, 2, 3, 4, 5 petals. This one on the bottom has 4 petals, right? 4 petals. Hence the 4s and 5s. There's too many stamens to really look at on these, but if you counted all the stamens, there would also be multiples of 4s and 5s in there, right? So for example, this might have 20 stamens. So when they say the 4s and 5s thing, it means It could be multiples as well. Like, so for example, this one has 5 petals, but it could have 10 petals, or it could have 20 petals. Those are multiples of 4s and 5s, and so we know it's a eudicot. Whereas monocots have flower parts in 3s. So on this lily, remember on lilies, the sepals look like petals, but just trust me, here's 1 petal, 1 petal, 1 petal. So 3 petals, 1 petal, or 1 sepal, 1 sepal, 1 sepal, 3 sepals. And it's easier to count the stamens here. 6 stamens, right? Multiple of 3. And if you were to dissect the flower, the parts of the pistil would also be in a multiple of 3. So eudicots have flower parts or whorls in 4s and 5s. Monocots have flower parts or whorls in 3s. So that's one way you can tell them apart. Another way to tell them apart is you can look at the pollen grains under the microscope. So the eudicots have pollen grains that have 3 openings. I'll use yellow. So on the top left here, you see a close-up of a pollen grain under the microscope. This pollen grain has 3 holes in it, 3 openings, and that's a characteristic of the eudicots. However, monocots have pollen grains with a single opening. And so that's another way you can tell them apart. Oh yeah, and just so you know though, like, pollen is very diverse as well. Like obviously on the previous slide, it's like, you know, 3 openings versus 1 opening, but depending on the plant you're looking at, there's a lot of diversity in pollen grain shape. So on the bottom here is actually SEM microscope picture of different pollen grains. You can see a lot of different sizes, a lot of different shapes. Some have pokey things, some don't. Right, let's look at another slide. So this isn't a slide, this is a collection of slides together, but check out these types of pollen. Like look at the different shapes, super cool. Here, like this one, like look at, like this one almost looks like a spaceship, right? Crazy. Look, this one looks like some scary lizard, like from a movie. Right, this one kind of looks like a cookie. But wait a minute, hold on. That is suspect. That is Mickey Mouse. Mickey Mouse, not pollen from an angiosperm, that's pollen from a gymnosperm. Get out of here. Right, okay. Vivian says, I was gonna say water molecule. No, that's Mickey. Remember Mickey Mouse? Mickey Mouse, remember from the gymnosperm, right? You got the pollen, you got the air bladders here, right? You could even do like this. Hi guys. I can't do Mickey voice. But yeah, get out of here. Not from an angiosperm, that's from a gymnosperm. But all the rest here are from angiosperms, and you could just see like a ton of diversity in shape and size and color. This one looks like some olive, you know, like just this one gives me nightmares, I will say. It's like some evil lizard monster. All right, I'm exposing myself. Let's move on. All right, pollen. Yes, so pollen's super cool because pollen fossilizes very well. So you might think, like, when we were looking at things in the fossil record, yeah, we're missing a lot because a lot of time plants are very fleshy. And so depending on what kind of ground they're growing on in evolutionary time, they might not fossilize very well. However, pollen does. Pollen fossilizes super well. And what's cool about that is pollen can really tell you about ancient environments and even some plant and animal interactions across evolutionary time. So for example, you can find pollen in fossilized dung, which fossilized dung has its own word, which is coprolite, just so you know. Yeah, somebody made up a word for fossilized dung at some point. Yeah, but you can like look at fossilized dung. Dung is poop, just so you know, dung is poop, but fossilized poop. and you can see the different pollen grains in there. And then you have an idea, like if you know that fossilized poop came from like a giant sloth, right, in South America, like a giant sloth that was like 9 feet tall, and you find some fossilized giant sloth dung, and there were certain pollen grains in it, you could see like, oh, this giant sloth existed at the same time that this plant existed, and they existed together in the same environment. If you want to learn more about fossilized dung, which I know you do, I know you do. We don't have time to do it right here, but there is a link here, and you can learn more about what corpylites are. Yes. Believe it or not, you could actually spend your whole life studying pollen fossils. So a person who does that is a palynologist. Yes, that is a job, palynologist. And this is a person who studies pollen, exclusively studies pollen, including pollen fossils. So here is Dr. Berkovici working in North Dakota, where they actually saw in those fossils that there were palm tree, look, yeah, example, palm tree pollen in North Dakota, which you really wouldn't think of North Dakota as a place that palm trees would grow, but they did at some point when it was a lot warmer there over evolutionary, geological time. So it could tell us different things about the environment over time, since things changed over time. Okay. Right. Oh yeah, another thing that we have learned from pollen fossils is also palm trees on Antarctica. Yeah, so we've actually been able to do cores there as well. And so remember back when we were first learning about, I think it was the Carboniferous forest back in the lycopods, the continents have moved over time, right? And during those times, things like Antarctica that didn't always used to be at the South Pole, right? It used to be other places. And when it was of those other places, at some point, there was actually palm trees on it. If you want to learn more about that, you can see the full story at the bbc.com link at the bottom of the slide. All right. So, so far from monocots and dicots, we've learned that you can tell them apart in different ways, right? One way is number of flower parts, right? The eudicots have flower parts or whorls in 4s and 5s. Monocots have flower parts or whorls in 3s, right? Another way we learned already is that you can look at the pollen grains. The eudicots have pollen grains with 3 openings. The monocots have pollen grains with 1 opening, right? So those are ways that we've learned so far. The other way you can tell them apart is the seeds. So, and in particular, the main difference between the monocots and the eudicots is the number of cotyledons or embryonic leaves. And that, if you know your Greek and Latin roots, that makes sense because look, cotyledon is an embryonic leaf. Cotyledon is how you say that. And mono and eudi, but then what do they end with, right? What do they end with, these words? They end with cots, right? And those cots refer to cotyledons. So you might even guess, right? What does mono mean usually? Maybe we haven't covered that. I don't think we have maybe covered mono, but mono means 1. Yes, thank you, Mary. Thank you, 1, right? Thank you, Catherine. So mono means 1. Cotyledon refers to, or cot refers to cotyledon, and so monocots have 1 cotyledon. That's why they're named that. And remember, it used to be dicots, right? Dicots, now eudicots. But either way, we know what di means, right? We know what di means, 2, and the cots refers to cotyledons, and so eudicots have 2 cotyledons. That's another way to tell them apart. Let's go here. Here we go. Here is a cross section through a eudicot seed. And it's kind of hard to see here. Actually, it's not. Never mind. It's not hard to see. This seed has 2 cotyledons. I will show you where they are. Here is one. Here is the other. If you've ever cooked beans or eaten beans, you can think of like a lima bean or even a baked bean or a pinto bean or any bean really that most beans, they kind of will split in half, right? Like you can split them in half. Another example could be like a peanut. If you've ever eaten like a whole peanut, when you take out that part, it will split in half, right? Well, the split in half is the 2 cotyledons. And those are the embryonic leaves. Cotyledons are the embryonic leaves, not to be confused with the foliage leaves, which will be the first mature leaves. Don't get confused. Cotyledons are embryonic leaves, not the same thing as mature leaves that will grow. Right, cotyledons. Whereas the monocots only have 1 cotyledon. So for example, like corn. Are they the, all right, so question about cotyledons. Are they the ones that make the initial sprout? Yes, they are. We will learn more about what the cotyledons look like in real life, either in today's lecture or in Thursday's lecture. But yeah, they serve a couple purposes, but they are the first leaves to emerge, but they look completely different from the actual mature leaves that come out. Yeah, but they are the first to emerge. Opposite from something like a peanut, which splits in half, right? Or a lima bean that splits in half, monocot seeds don't do that. So for example, corn. So if you've ever taken a kernel of corn, right? Or if you've ever made popcorn, and you've seen the little popcorn kernels, those don't split in half, right? Those are a single thing. Like good luck trying to cut a popcorn in half. Don't do that. You get hurt. But it's a single thing, right? That's 1 cotyledon. And that's because corn is an example of monocot. Monocots have a single cotyledon. Yes. Don't worry about the terms on this slide. The point is grasses are weird. The point is 1 cotyledon. It's not labeled on this one, but like this is corn. And basically, the point is 1 cotyledon, 1 cotyledon. In the case here on this onion, it's this thing. The corn, it may be passed on this slide. It doesn't matter. Okay. The point is number of cotyledons. Dicots have 2, monocots have 1. That's the point. That's the point. Oh, right there, I said at the bottom, right? In grasses, the cotyledon is called scutellum, right? Because it's shaped like a shield. Thanks for being complicated, grasses, right? That's why I'm like, don't even worry about this. Thanks, grasses. Way to be complicated. Yeah, but the point is 1 for the monocot, right? You don't need to know scutellum. Just show you where it is. All right. So I like to do this, keep you organized. How do we tell monocots and dicots apart? Monocots have flowers arranged in 3s. Dicots have flowers arranged in 4s and 5s, right? Monocots have pollen grains with a single opening. Dicots have pollen grains with 3 openings. Monocots have 1 cot, right? 1 cotyledon. Dicots have 2 cotyledons, right? Dicot. Another way to tell them apart is their venation, right? What their veins look like in the leaves. So eudicots have what's called a net-like or reticulate pattern of venation, which means that the veins are kind of like almost like a net. Maybe it's not super clear here, but normally what I like to think of to try to keep that in mind is like normally with a net, like you have a single main vein and like other veins coming off of it, right? that tends to be your net-like, right? We have a single main vein and then all these others, but like all these other little things are also veins. Sometimes the main vein might not be as obvious, like this one has a main vein there, and then there's other ones coming off and like other little ones coming off. And then you may have some variations on that. Sorry, I don't know if you can hear that thing. I'm going to turn it off. You might have variations on that where it's not as clear, right? Like, so for this maple leaf down here, it's kind of like not really like a main vein, but all of these veins coming out of this one spot. Similarly to this, but the point is, it's kind of net-like. Maybe you're looking at that and saying that doesn't look net-like at all. Well, maybe it'll be more clear once we get to the monocots. Monocots don't have this net-like thing. Monocots don't. Monocots have parallel veins, parallel venation, and that the veins run parallel to each other. So it's very clear here in B, all of these lines, I'm going to try to draw straight. This is not going well already, but all of these things are running parallel to each other. Those are the veins. Here, you can see that the veins are running parallel to each other. Likewise here, parallel to each other. So that's another way to tell them apart. Monocots have parallel venation. Dicots have this like net-like venation. Okay. You can actually, so you can see this with your eyes. If you're walking around and you pick up a leaf, you can look at the leaf and you could say, oh, this leaf has parallel veins. This leaf must be from a monocot, right? You can see this with your eye, but you could also see this under the microscope slide. You might see this microscope slide in lab, just so you know. Yeah, so you can see these differences under the microscope slide as well. Here you go. So on the left, let me just move this box. I'm gonna move this box out of the way. On the left here, you have a eudicot leaf. And on the right, you have a monocot leaf. And so what you want to look for is the veins. Sorry, I thought we had a question. What you want to look for is the veins. So for the veins on the monocot leaf, which is on the right, come on, right. So it's color coded for your convenience. Monocot leaves are going to be coming, monocot veins in leaves are going to be coming at you if you do a cross section because they're all parallel. Whereas the dicot leaves, you're not going to see the veins coming at you in the same way. One way you could think of it is this way. Here, I'm going to stop share for a second just so I can kind of show you guys. So like monocot leaves have parallel veins, right? So like this, you could think of this as like a monocot leaf, right? And you can think of my fingers as like the parallel veins, right? Parallel veins. If we're going to do a cross section through this leaf and you're going to look at it this way, right? Like a cross section, the veins are going to be coming at you. They're going to be coming at you because they run parallel, right? So if you do a cross section through here, all the veins are going to be facing you like the tops of straws. Whereas if you're a net-like leaf, a leaf with net-like veins, right? And you do a cross section through this, sure, like you might, one of these might be coming at you, right? Like, so maybe like here, like this one is coming directly at you, or like this one is coming directly at you. But like the ones next to it, you're not going to see them like straight on. They're going to be kind of like the sides of it, things like that. So that's what this is trying to show you here. Yes. So like on the monocot leaf on the right, oops, come on. Those red circles are each showing you a vein, and inside of those veins, you can very clearly see the xylem and the phloem because it's like you're seeing the complete vein. Whereas in the dicot leaf on the left, the one that's in the circle, you can see the full xylem and phloem, but the one that's in the rectangle, you're only seeing a side of it because since they're net-like, they're not all going to be coming at you straight on. Okay. All right, yet another way we can tell these guys apart. So another thing that they have different is the way that their vascular bundles are arranged in the stem. So on the left here, you see a Dicot stem cross-section. And on the right, you see Monocot stem cross-section. And in the Dicots, the vascular bundles, let me change colors here, are arranged in a ring. So like here's a vascular bundle, here's a vascular bundle, here's a vascular bundle, etc. And all of these vascular bundles are arranged in a ring for the Dicots. If we do a cross section through monocot though, each of these dots is a vascular bundle. Like here's one, here's one, here's one, etc. And obviously those are not arranged in a ring. They are scattered. They are scattered everywhere. You guys will also come across this in lab. So ways to tell them apart: number of flower parts, right? Number of pollen grain openings, the way that the veins are arranged, the way that the vascular bundles are arranged in the stem, the number of cotyledons, right? These are all ways to tell them apart. And if you look in the root, you have another difference in that the Dicots roots, and again, you guys will cover this in lab as well. If you do a cross section through a Dicot root, there's only one vascular cylinder and it's right in the middle. But if you do a cross section for a monocot root, there's still only one vascular cylinder, but it's a lot bigger and it kind of almost appears arranged in a ring. So there's these parts of the vascular cylinder, don't need to know them today, but they kind of look like they're arranged in a ring. Whereas if you look at the vascular cylinder in the Dicot root, it's almost like X marks the spot. This is how I visualize these two. And you might want to as well. It's helpful. All right. So those are the different ways. Oh yeah. Another thing is that Dicots have true secondary xylem, right? That comes about via the vascular cambium, whereas monocots rarely do. Monocots, remember, I said before, monocots are things like grasses and bananas and things like that, bamboo. Those don't really get super thick like a tree, right? They don't really get super thick, so they don't really have that much, they don't really make secondary xylem as we think of it. Sometimes they make a little bit, but when you think of like a woody tree, you're almost always talking about a Dicot. Whereas look at the difference of this. I think this is sugarcane on the right, which is a type of grass. Yeah, not woody. Okay, here's a nice summary table for monocots and Dicots that kind of tells you all the things that I just covered, right? So nice summary table. And then as promised, this is that summary table that I mentioned about xylem and phloem of the tracheary elements and the sieve elements and the other cell types and what they, to keep them organized in your head. And just in case this, you might find this helpful, you might not find this helpful, but this is another summary table of all different cell types. Things like parenchyma, sclerenchyma, that might be something that is useful in lab, but this is just for your reference, right? Just a summary slide of different cell types. Okay, all right. So
can I ask a question?
Yeah, and we're not done with this. We're going to start the next section, but yeah, go for it.
Okay, the basal angiosperms, the rest of the classes.
Yeah.
How would you characterize those? Are those just like a combination of the ones we've discussed?
Mm-hmm. Yes,
okay.
That's why they're like. That's why before, when we had just monocots and dicots, it was like we really had a handle on what a monocot was back then. Like we knew a monocot had 3 flowers arranged in 3s. We knew that they typically weren't woody. They only had a single opening in their pollen grain. And then like all the other plants that didn't fit those criteria, we kind of like lumped them together because they were like, they're not monocots, so they're something else. And for whatever reason, people were really hyper focused on the number of cotyledons, like such that it was like the thing that named the 2. But as you just mentioned, yeah, so the basal angiosperms sometimes have a combo, right? So like maybe their flower parts are arranged in 3s, like a monocot, but maybe their pollen grain has 3 openings, like a eudicot, or maybe the way that the vasculature inside of the stem looks like a eudicot, but it only has 1 cotyledon. Yeah, they're funky. They're funky, and we don't cover them in this class just for that reason, because they're just too different, too diverse. Good question though.
Sounds like not such a perfect classification.
Right. Well, that's why. So like, spoiler alert, yes. That's kind of how it is in the sciences in general, particularly in the plants, because, you know, as humans, we really want to put things in neat little boxes. I don't know why,
Right.
but we always want to do that. And you have to imagine here, there's 366,000 plants that make flowers that we know of. And so there's always going to be an exception to the rule, because there's just too many. You would have to make, every plant is a little bit unique in its own way. So you'd have to make 366,000 boxes.
Right. I was wondering, I was thinking like monocots and eudicots were within themselves related to each other or came from a common ancestor, but now like with the confusion of basal angiosperms, I don't even know if I could say that.
Right. Well, and that's kind of correct. Yeah, because look, if I pull this back up here, let me go back to that slide real quick. Here, yeah. So let me share this. Boy, I have so many boxes. I don't even know where you guys are. Here you are. Okay. We'll go here and we'll share this. Yeah, so like in some ways, the basal angiosperms are, we say basal because they are, here, let me, this isn't always the case. Like I just said, like there's always exceptions, but when we say basal, we're referring to the fact that they are closer to they're more closely related to the common ancestor. They're toward the basal part of the tree, which means like the bottom part of the tree. So things like this are more, they're kind of like on their way to becoming either a monocot or a dicot. That's one way to kind of think about it. That's why they might have different combinations of these things, because they are older in evolutionary time and kind of like on their way to, yeah. But yeah, monocots and dicots are still very good distinctions for like discussing plants. Like they do have these things. Basal angiosperms definitely make the angiosperms in general more complicated, but that's why we don't talk about them really in this class. Yeah, good questions though, Catherine. All right, let me share the next one. We're going to start this one today because we have to finish it. We have to finish this stuff today and Thursday. So we are going to start this today. We're obviously not going to get through all of it, but we will begin it. So this slides are the last slides of the course, half of which we'll probably do today, half of which we'll do next time. So we already talked about this, right? And we know that all the angiosperms make flowers, and we know that the flower is a specialized shoot system. However, the leaves now, what used to be leaves of this specialized shoot system are now flower parts that are modified to do reproduction, right? And there are different sex structures, male sex structures and female sex structures. And I think we talked about this before, but yeah, the stamens are the male structures, and the pistils / carpels are the female structures, right? You could also use those more fancy words, androsum and gynosium, if you prefer. This is a recall slide. All right, now we're going to talk a little bit about flower form and how different flowers are arranged on plants. So some plants have, and this stuff that I'm about to cover will be really useful for you if you're ever walking around and you see a plant with a flower on it and you want to figure out what kind of plant is this. You're learning different ways to describe these plants, right? Like you could look at the number of flower parts. If you were able to cut part of it and look at the interior anatomy of the stem, things like that, right? If there were seeds present, you could look at the number of cotyledons. And this could help you identify what plant you're actually looking at. So the way the flowers are arranged are another way that you can kind of identify plants. So some plants have flowers that are solitary, or AKA born singly. For example, this rose, right? This rose, solitary flowers. Here's a stem, here's one flower, right? So that's okay, comfortable with that. Other plants have groups of flowers that are together. And when that's the case, we call that whole thing an inflorescence. Inflorescence. So in the case of this agapanthus right here, here's a stem. And then each of these little things are a flower. So this is a group of flowers, hence an inflorescence. This whole thing is an inflorescence. This whole thing is a solitary or born flower that's born singly. So that's one way flowers can be arranged. Look, just because we just went through this, look, this flower has flower parts arranged in 1, 2, 3, 4, 5, 6. 6 is a multiple of 3. So what must this plant be? Mono. Thank you, Roseman. Yes, monocot. Right? Good. See, so now you could already walk around and be like monocot, dicot, monocot, dicot. All right, good. All right, next. Oh yeah. So if you're a stalk that's bearing a single flower, we call that stalk a pedicel. If you're a stalk bearing an inflorescence, it's called a peduncle, which is a term we've already come across in another context in this class. But just so you know, All right, but there's a lot of different types of inflorescences. Inflorescences, right? So remember, an inflorescence is a group of flowers, and there's a whole bunch of different types. Here is a summary slide of the different types. And we're going to go through a few of them right now, just to help you understand these arrangements. So on this slide and the subsequent slides, every time you see a little circle, a little red circle, that indicates a single flower. So each little circle is a single flower. Here is an example of an inflorescence that we call a panicle. Panicle has, it's kind of like the parts of the panicle, they're kind of like alternately arranged, these little stems that come off. And then at the tips of all the stems are little clumps of flowers, right? So one example of that is this hydrangea here on the right. So that's called a panicle. Another type of inflorescence is a spike, as shown here on these lupine. Lupine is the type of plant that we learned about in that documentary way back. But, you know, spike has a single stalk and then little flowers coming out of the stalk. So that's a spike. If the little stick drawing is not helpful to you guys, there's usually also a more botanically accurate drawing as well on these slides. So a spike. Then you have a raceme. Raceme is kind of like a panicle. Let's go back. Kind of like a panicle in that it has a single stalk with little stalks coming off of it. But the difference is there's only one flower on the tips of all these stalks, right? Here's one flower. As versus the panicle, which has groups, little clusters of flowers, right? So like on this little stalk, there's a cluster of flowers and not one, right? So that's the difference between a panicle and a raceme. Another type is a quorum. Quorum is kind of easy to identify because it almost looks like a upside down, it kind of looks like a triangle. So like here you have the stalk, and each of the little stalks are going to be different lengths such that all the flowers will be at the same height, right? So like this stalk is super long, this stalk is kind of long too, but this one is a little shorter, and it gets shorter and shorter until all of the different flowers are at the same height, right? You can see here. And quorums are nice because it kind of serves as like a big landing pad for pollinators. Next is a simple umbel. Umbel is similar to a quorum. I got a question in the box. Looks like broccoli. Yeah. It does. Don't. It's not though. Yeah. Quorum and umbel are similar in that, like, they're kind of similar. And the difference is that in the quorum, the stalks are different lengths, right? Such that all the flowers end up at the same height. In the umbel, the stalks are all the same length, radiating out of one spot. And in some cases, you might end up with the flowers at the same height. Other times, the umbel might look like a big ball. Simple umbel, and then because plants are complicated, you also have compound umbel. Compound umbel is like an umbel of umbels. That's how you can think of it. Like, so like here is one umbel, right? Here's an umbel. And on a compound umbel, instead of having a flower at the tips, you'll have another umbel. Like this. I think compound umbels are super cool. Just like very, very interesting, pretty flower, or sorry, inflorescence shape. Queen Anne's lace on the right here is an example of a compound umbel. A lot of things in the parsley family, so carrots, parsley, hemlock, obviously Queen Anne's lace, those all have this, celery have this type of a flower, inflorescence, right? Compound umbel. So an umbel of umbels on the tips of each of these stalks is another umbel, right? So an umbel of umbels, or a compound umbel. So these are just different types of inflorescences so that when you're walking around and looking at plants, you can identify what you're looking at. A lot of plants right now, and maybe during your phenology assignment, you encountered this, have an inflorescence type that's called a catkin. Catkin is a stalk that hangs down with little flowers all over it. Here's one here. Kind of looks like a strobilus. I'm not gonna lie. Kind of looks like a strobilus, but it's not. It's a catkin because it's not. Remember, strobilus is a stalk with leaves on it. In this case, this is a stalk with tiny flowers on it. But from far away, yeah, kind of looks like a strobilus. But if you were to examine it close up, you would see that there's tons and tons of little flowers all over this. Birch has this, willow has this. Lot of trees right now probably have this on it right now. Next is a head, or AKA a composite flower. It's called a composite flower because it's composed of two different types of flowers. It should be called composite inflorescence. Because it's a inflorescence composed of two different types of flowers, disc flowers and ray flowers. Your sunflower is an example. Catherine, what do you hate? Sunflowers? No. Catkins? The catkins, okay. I figured it wasn't sunflowers. Yeah, so sunflower is a good example of a composite flower, or AKA a head. Two different types of flowers on a sunflower. I'll use, I don't know, pink. I'll use pink. The middle part has what are called disc flowers, which are shown nicely here. Well, actually, which are shown nicely in the middle here. And then each of these things coming out is another type of flower that's called a ray flower, which would be these guys here, right? Here's your disc flowers here. Whoa, sorry. Disc flowers in the middle, and then each of these yellow things is the ray flower. And I always say this, I have to say it, I always say it. We call this thing a sunflower, right? We call this thing a sunflower, but we really should be calling it a sun inflorescence, not a sunflower, because it's an inflorescence. It's a structure with multiple types of flowers on it, more than one flower. Sun inflorescence is what it should be called, but fine, sunflower, fine. Easier to say, but it should be sun inflorescence. Okay, just in case you don't believe me that there's different types of flowers there are. Here's a cross section, sorry, longitudinal section through a sunflower, sun inflorescence, showing you the disc flowers in the middle and the ray flowers. Sorry, I'm just refilling my water, parched. Disc flowers in the middle, ray flowers on the outside of this sun inflorescence. All right, how are we doing on time? All right. So the rays have male or female parts. Oops, did not mean to do that. Let's go back. Good question. So if you can see here, so I should say, I think the ray flowers, I'm not 100% sure here, but I'm pretty sure. I think the ray flowers don't have, Ben, correct me if I'm wrong here, but I think the ray flowers don't have either. Maybe they have, I think the ray flowers are mainly just a petal at this point, and maybe the remainder of a sepal. Their job really is to signal pollinators. I can tell by this diagram here that the disc flowers, you see these little branch, like this branched thing coming out the top, that's 100% a stigma of a pistil. So I know from that the disc flowers for sure have the female parts. It could be that the male parts are these little things at the bottom. So if I had to guess based on this drawing, I would say that the disc flowers have the male and female sex structures, and that the ray flowers don't have any either anymore and are just now a petal. But correct me if I'm wrong, if anybody has more information about that. Okay, so I'm gonna see, let's just see how I'm doing here. Where would be a good place to stop? I think we can at least start, this and then we'll see how we go. We might have to pick it up. Obviously, we're gonna have to pick it up on Thursday. So angiosperms can be monoecious or dioecious. We have learned what monoecious or dioecious means before. Monoecious means in one house. Dioecious means one house. Remember, don't get confused here. Monoecious and dioecious refers to the plant, not the flower. Monoecious refers to the plant. Dioecious refers to the plant. So if you're a monoecious plant, on your plant body, you'll have either perfect or imperfect flowers, but both types. If you're a dioecious plant, you can only have imperfect flowers, period. Because an imperfect flower, go back to those slides that give you the definitions of that. But an imperfect flower is missing one of the sex organs, right? Either the male or the female sex organs. So if you're a dioecious plant, you can only have imperfect flowers, because if you had perfect flowers on you, you would have both sex structures on you, and then you would be a monoecious plant. So when you're thinking of monoecious and dioecious, remember that you're talking about a plant. If you're wanting to discuss the flowers, you have to say either perfect or imperfect to discuss the flowers on that plant. Yes, just a recall slide telling you the parts that are the male and the female structures. Okay, here is an example of perfect flowers on the left and imperfect flowers on the right, right? So for perfect flowers, you have both the male and female sex structures on one flower. I'll use green. Here is the top of the female sex structures, and then all of these things surrounding that are the male structures on this flower. But on the right here, you have examples of imperfect flowers. This is a flower here that only has the female sex structures. And then this is a inflorescence here that has only on it, but you can say flower for now. Has on it only male sex structures. So two different flowers, perfect versus imperfect flowers. Okay. So we're going to start angiosperm reproduction day. We're probably not going to finish it. That's all right. We'll catch up next time. But for the sake of simplicity, we're going to go through the steps using an example of a perfect flower, okay? Because it's just easier to discuss the steps of reproduction using a perfect flower example. Question, should I be able to tell from this photo if it's pistillate or staminate? It's hard to tell. No, not necessarily. Like, I'm just giving you an example of, yeah, of the two. Okay. All right, so let's talk about angiosperm reproduction. So here we go. We have our perfect flower. We're going to use as an example. And the perfect flower has on it male and female sex structures, right? The left bottom, we see the female sex structures. On the right bottom, we see the male sex structures. first, let's talk about the female sex structures, right? And yeah, first let's talk about the female sex structures, aka pistil, aka carpels, aka gynosium, but whichever you prefer. What are the parts of the female sex structures? There's a stigma. Oh, so sorry. So you see the red box. The red box is showing you all of the things. So the red box is showing you the pistil, aka carpels, right? The whole thing. Next, you have, if you were to zoom in that pistil, these are the different parts. So at the top of the pistil, you have a stigma, which catches pollen, right? Sticky stigma. And stigmas have a lot of diversity. These are different examples of stigmas. Look at this one, like curlicue. This one's like a pom-pom. This stigma almost looks like a big tongue, right? You get branching sometimes, little frillies sometimes. So all different types of stigmas, but doesn't matter what they look like. The point is that their job is to be sticky and catch pollen, and they do, sticky and catch pollen. Here is a close, or here's actually pictures of different types of stigmas. So look, here's our curlicue again, right? This one here is branched, right? This one here has 2 branches. We got little pom-poms here. So all of these are examples of stigmas that are sticky. And you could actually see on some of these, you could actually see pollen grains. Look, see those little dots that are on there? Those are pollen grains. If you were to look close up here, there's some orangey stuff. Those are pollen grains on there. So sticky stigma catches the pollen. Next, you have the style, which is this thing that extends the stigma out of the flower. And it's solid. The style is solid, not hollow. It's not a tube. And then lastly, you have an ovary. And inside of that ovary is 1 or more ovules, depending on what plant you're talking about. There's your ovule, right? You can see a total of 4 ovules in this drawing. All right, remember, with the angiosperms, protection is key, right? The ovules in the angiosperms are better protected than in the gymnosperms because the ovules inside of an angiosperm are inside of this ovary, right, which is this here. And each ovary is composed, in the case of the angiosperms, of 2 integuments. And this is why I want you guys, if you're interested, and also for this extra credit, I think there will be more discussion about this at the Torrey Botanical Society lecture that's held on Monday. So I'm interested to see what they talk about, or what's talked about there in terms of integuments. But the point is that ovules are better protected, right? They're better protected. They're inside of a structure called an ovary, and the ovary walls have 2 integuments. Oh, sorry. I got carried away. Started thinking about this lecture. This is the ovary. Sorry. I got excited. And then look, here's your integuments in here, right? 2 of them. And so the ovule is much better protected than we were in the gymnosperms, which is on the left. Yeah, so here we go. We're going to talk about ovules a little bit more in detail for the angiosperm ovules. So what is in an angiosperm ovule? There's a bunch of new words here, but we will encounter them today and next class. So inside of an angiosperm ovule is a female gametophyte, but it is so reduced. And remember, over the course of this class, like all of the classes, I've really tried to emphasize the fact that as plants become more evolved, the gametophyte plants become more and more reduced, right? More and more reduced. In the case of the angiosperms, the female gametophyte plant has become so reduced that it only consists of these, what is it, 4, 5, 6, 7, 8 components. There's only 8 components now that it's made up of. There are these 3 antipodal cells. Do I highlight them? Let's see. Yeah, no. 3 antipodal cells, which are up here. 2 synergid cells. Here's one. Here's another. 2 polar nuclei located there. And 1 egg there. And that's it. That's the entire female gametophyte plant. Super, super reduced. It is so reduced that it is now microscopic. Has to be seen with a microscope. It is super, super, super small because it has to fit inside of flowers, right? It has to fit inside of flowers. Yeah, and unfortunately, in the angiosperms, the female gametophyte is now called the embryo sac. Like, remember pollen sac, right? Now we have pollen sac and embryo sac. I don't really like the term embryo sac, but that is indeed what it's called. Okay. I don't think, oh boy. Yeah, I don't think I'm going to go into megasporogenesis. Yeah, we'll pick up with megasporogenesis next time because it's too much to go through too quickly. And then the rest of this slideshow, there's like pictures and videos and things like that. So we'll have time to go through the megasporogenesis next time. It's just too much. So yeah, and I know some of you have class directly after this, so I don't want to keep you. So yeah, I thank you guys for joining me today. Hopefully we're able to get through the rest of the material on Thursday. Next week, we have special guests, which I'm super excited about. I hope you are as well. And yeah, good luck with all the rest of your day and the rest of your classes. And I will see you guys on Thursday. If anyone has any questions, happy to answer them here. Otherwise, have a great day, guys. It's hard to talk for this long. Don't recommend it.
Professor.
Bye.
Professor,
Yeah, Brittany, what's up?
when you said if you were to dissect the monocot, it has rows of 3s, and you said the pistils in 3s, I only imagined it as 1.
Yeah, here. What's the best way? Right. So it's what is the best way to there is, so for the purposes of this class, there is only 1 pistil, but there might be different numbers of, here, I think I know what will help. There might be different numbers of chambers inside of the ovary. Here, let's go to probably the other slideshow, show this better. Pull this up here. Let me go. Here, 1 second. No, that's not useful. Let me pull up the other one. Open, let's try this, part 1. Yeah, okay. Sorry, Brittany, just give me 1 second. Okay, here we go. Okay. Yes, I'm gonna share this with you. Let me see here, share screen, and we'll go here, share. All right, can you see this slide, Brittany, with the whorls?
Yes.
Okay, great. So you're right. I'll make it big first. It is like a single pistil, like pistil is a term for like the whole middle part, but sometimes sometimes there will be different numbers of, so this is not a great, this is not a great example because 1, 2, 3, 4, 5, 6, right. This is a made up flower. Don't, this is just for the purposes of learning whorls. But for example, inside of this pistil, this is one pistil here. You have 1, 2, 3, 4 ovules that you can see. And so when we talk about those things like arranged in 3s and 4s, or sorry, 4s and 5s, or arranged in 3s, it might be like the chambers of the pistil. So like on this pistil, there's like 2 chambers. So in a monocot, again, this is a made up flower, just trying to show you whorls, but like in a monocot, you might expect that there would be, if there were chambers inside of the pistil, there would be 3 or 6 chambers, right? Or maybe there would be 3 ovules or 6 ovules or 9 ovules in that pistil. Whereas in a eudicot, if your pistil was divided into chambers, you would have 4 or 5 chambers, or multiples of 4 or 5. Maybe you'll have 8 chambers, or 12 chambers, or 16 chambers, or number of ovules, right? You might have 4 ovules, or 8 ovules, or 5 ovules, or 10 ovules. Does that make more sense?
That makes sense. Thank you, Professor.
No problem. Glad to help. Have a good
great-
one. Bye.